Alexander. I'm the Product Application Scientist for IDseq, and today I'll be going over how to build a consensus genome in IDseq, how to make sure it's a high-quality consensus genome, and then a little bit about some of the output files that we provide. And I'll also be going over the pipeline. And I just want to say that the pipeline was first developed by the Zuckerberg Biohub. So we have collaborated with them on this project. So the first step is to remove the host reads. So once the raw reads are uploaded onto IDseq, they are aligned to a host or human reference genome and removed from there. And then the rest of the reads are aligned to a SARS-CoV-2 reference genome. We use the same one as NextStream. And this is done by using Minimap. Then those reads that align to the reference genome are run through a Kraken database. This is a little bit of an odd step for building a consensus genome, but because we originally had built this pipeline for MSSPE, this step is run to remove any reads that are homologous to SARS-CoV-2 that are not SARS-CoV-2. We also have human reads in this database, so if there's any leftover human reads that were not removed during the first step, they're removed during this step. From there, low-quality reads are removed and adapters are removed. Then the reads are aligned back to the SARS-CoV-2 reference genome by Minimap. IVAR is used to soft clip the primers, and then the consensus genome is called and the variants are called. And so we require 10 or more reads to align to a specific location for the consensus genome to be called, and a base must be at 90% frequency for that base to be called. Okay, so I'll go over how you would upload this onto IDseq. So if you go to idseq.net and log in, you can go to our upload tab, and then you can either create a project or upload to an existing project, and then you'll choose your analysis type. So if you want to make a consensus genome, you'll choose the SARS-CoV-2 consensus genome, and then from this drop-down menu, you'll pick which wet lab protocol you used. So right now we only support ARTIC P3 amplicon sequencing and MSSPE. And once you choose that, it will inform the pipeline which primers to trim. Then you can upload from your computer or directly from BaseSpace if you have a BaseSpace account. So from here, you can add the metadata associated with the sample. So you can add human here if it's from a human host. Add your sample type. So if it's nasopharyngeal swabs, you can add that here. And if you have water controls, you can toggle this on to identify them as water controls. You add your nucleotide type, so RNA, collection date, and collection location. And then from here, you upload, and then the pipeline will run automatically in the cloud. It usually takes less than 15 minutes for it to complete. And all the code available for the pipeline is available on GitHub. So once the pipeline's done, you can kind of dig into your results. So this is what your results page will look like. You'll have some metrics to run some quality control checks and a coverage visualization. So the coverage plot is actually a great first QC check to see if you have the full genome or if there's a lot of gaps. So along the y-axis here is referred to as depth. So how many reads aligned in a specific location. And then across the x-axis is coverage breadth. So how many reads aligned across the genome. And for any of the consensus genomes to be called, there must be at least 10 reads in a specific location. So this looks like this has the full consensus genome. There's really nice coverage across the entire reference genome. Well, this one, there's lots of gaps, and while there are some places where reads have aligned, nothing will be called in these locations because the depth is below 10 reads. So once you've determined that you have the full consensus genome, or maybe you have pieces that you want to do some more quality control on what you do have, you can use these metrics at the top here. So we have the number of reads from your run and the GC content. Typically, the GC content, you want it to be around the same as the reference genome. So this looks good for SARS-CoV-2. It's usually in this range. And then the next step is to look at the number of SNPs. So SARS-CoV-2 is slow to evolve, so we really don't expect a lot of SNPs. So if you see 30 or more SNPs, it's a bit of a red flag, and you might want to dig into some of those SNPs a little further. Also, we provide the number of informative bases. So NextStrain requires 92% of the reference genome to be covered. So that translates to about 27,510 informative nucleotides. So A's, T's, C's, and G's. So this one would pass that threshold if the goal was to get it onto NextStrain. And then missing bases is the number of N's. And ambiguous bases is if a base could not be called because it didn't meet that 90% threshold. So if there's 50% A's, 50% T's in a location, there'll be an ambiguous base in that spot to represent that it could be an A or a T. Okay, so then we also provide these output files with this download all button. And there is documentation for everything I'm going over in our help center, which I'll give an address to at the end. But these are the files that we provide, and the ones that are highlighted in this light blue color, these metrics are, like the important metrics, are available in the user interface, the ones I just went over. So you can interrogate these files if you'd like, but I'm going to go over the ones that are not highlighted. So if you happen to spike in with ERCC controls, you can look at the ERCC stats text file, and you can see the number of reads that are quantified here. And then we have these no host, so again, like no human reads files that are really useful if you want to upload this to the sequence read archive on NCBI. And then there are primertrim.bam.by files. And these are complementary files, so if you want to investigate the reads that have a line to the reference genome, you can look at them in Genius if you happen to have Genius or CLC, depending on what you have available, or IDseq, which is a free genome browser where you can upload these files, and then it'll show you, here's the reference genome on the bottom, and it'll show you where the SNPs are located at a read level visualization. And then, of course, the consensus genome is consensus.fa, so this is a FASTA, and that's the main file here. So another QC step that is really nice to do so you can visualize where the SNPs are is just to upload that FASTA file onto Nextclave. And it'll give you a little bit of QC metrics along with where the SNPs are located, and then it will put it in a clade. So that's a little bit about building a consensus genome in IDseq and how to do the QC analysis. If you have any questions, feel free to email us at help@idseq.net. And as I said, we have a help center, help.idseq.net, where there's documentation for everything I went over, and then some additional analyses if you're interested in doing some downstream analyses from building your consensus genome. I hope that was informative. Thank you.